Hello everyone. Welcome back to the channel of the Food Professor. Today's topic is chemical composition of cereal grains. So, let's start. The first component is carbohydrates. Approximately 80% of the cereal grain is composed of carbohydrates, NFE plus crude fiber. Only 3% to 5% of these carbohydrates are structural or fiber containing. The rest is constituted by reserve carbohydrates, mainly starch. The starch molecules are stored in granules located within the endosperm. The starch is a polymer of glucose units joined by A1,4 and 1,6 glycosidic bonds. The granules are packed with amylose and amylopectin molecules. Amylose is a linear chain devoid of A1,6 glycosidic bonds. The amylose forms a helix and is the main starch responsible for retrogradation. The amylose contains 1,500 glucose molecules and has an average molecular weight of 2.5 x 10 to the power of 5. The amylopectin, also called branched starchy, is similar to glycogen present in the liver and muscles of the human system. The glucose molecules are mainly linked by alpha-1,4 glycosidic bonds but also contain branches that occur when alpha-1 to 6 bonds forms. Only about 4% to 5% of the total glycosidic bonds are alpha-1,6. These glucose polymers have a molecular weight of 10%, 600,000 glucose units/amylopectin molecule, and structurally are divided into type A, B, and C chains.
The amylose and amylopectin are packed inside the starch granule in such a way that the granule shows crystallinity and concentric circles when viewed under the microscope. The crystallinity is clearly observed when native starch granules are viewed under a microscope equipped with polarized filters. The presence of a Maltese cross or birefringence indicates that the granule is native or undamaged. When the starch granules gelatinize due to heat treatment or suffer mechanical or enzymatic damage, they lose birefringence and are considered gelatinized. The gelatinization phenomenon is irreversible and is defined as the loss of the internal crystal structure of the granule. Most starch granules lose birefringence when exposed to temperatures of 68 C, 78 C. The starch of most cereals contains 75% amylopectin and 25% amylose. However, some cereals such as maize, wheat, rice, barley, and sorghum can contain from 95% up to 100% amylopectin and therefore are almost amylose-free. These cereals.
Sweet maize has been selected to produce high quantities of soluble sugars by inhibiting the enzymatic transformation of these sugars into starch in the maturing kernels. In regular varieties and hybrids, the maize cob is harvested before it reaches full maturity or when kernels are in the milk or dough stage. In this case, the corn cob should be stored under refrigeration in order to reduce the metabolic activities in starch synthesis. X component is fiber. Cereals are considered a good source of dietary fiber. Both insoluble and soluble dietary fibers have positive health benefits. The insoluble fiber increases the peristaltic movement or transit of the digested food throughout the gastrointestinal tract, increases the fecal bulk, and prevents constipation, hemorrhoids, diverticulosis, and colon cancer. The soluble fiber reduces blood cholesterol and lowers the sudden increase in blood glucose levels. Both types of fibers have the ability to bind bile acids, decreasing cholesterol. Insoluble dietary fiber reduces mineral bioavailability. Oats is the cereal that has a good ratio between insoluble and soluble fiber. The soluble fiber is particularly rich in S-glucans and arabinoxylans. The insoluble fiber is mainly formed of cellulose and lignin. These chemical components are mainly located in the glumes, lemma and palea, pericarp, and endosperm cell walls. Cellulose is the major building block of the cell wall and is usually associated with other structural components such as hemicelluloses and pectin. The cellulose may contain up to 10,000 SS1, 4-linked glucopyranose units. Unlike the other fibrous components, lignin is not carbohydrate. It is formed from cinamyl alcohols that first form phenylpropane units, which by further complex polymerization form lignin. The soluble fiber is mainly formed of hemicellulose, arabinoxylans, and B-glucans. Hemicellulose is branched polymer consisting of different sugar moieties, xylose, arabinose, galactose, gluconic acid, and glucose. The B glucans, composed entirely of glucose units, have affinity for water and are within the soluble dietary fiber fraction. 3 glucans readily ferment in the hindgut and are considered hypocholesterolemic. Arabinoxylans are heteropolysaccharides consisting predominantly of arabinose and xylose residues. The official method of fiber analysis for labeling purposes consists of emulating the pass of the food throughout the human gastrointestinal tract. Ground samples are treated with proteases and amylases and washed with solvents to remove protein, starch, and fat-soluble compounds. The residue obtained after filtration is the insoluble fiber, whereas the recovered filtrate that contains the soluble fiber is treated with warm alcohol. The alcohol precipitates soluble fiber that is recovered by filtration and gravimetrically measured after oven drying. Third component is proteins. The amount of protein present in cereal grains differs according to species and even within the same species due to genetics and environment. The protein concentration varies due to environmental conditions during grain development and maturation in the field. The factors that affect protein concentration the most are nitrogen fertilization and water availability during grain development. Oats and rice are the cereals with the highest and lowest protein contents, respectively. Among the different wheat classes, the soft wheats have been bred to contain the lowest protein content, therefore they contain the weakest gluten. Hard and durum wheats usually contain from 10.5% to 14% protein. The gluten of durum wheat is usually stronger but less extensible compared to the gluten of hard wheats. As in other cereals, the protein is distributed in the different anatomical parts of the grain. The germ and aleurone layers contain the highest concentration. However, since the starchy endosperm is the largest anatomical part, it contains approximately 70–80% of the total protein. Proteins are classified according to solubility. The water and weak salt solution soluble fractions are called albumins and globulins, respectively. These two fractions are mainly concentrated in the germ and are composed of enzymes, nucleoproteins, and glycoproteins. These proteins are considered biologically active and play an important role during grain germination. Nutritionally, these proteins have the best amino acid balance and quality because they are well digested and contain high amounts of lysine and other essential amino acids. Among cereals, oats contain the highest amounts of these protein fractions. Approximately 80% of the cereal proteins are considered as reserve or storage. In most cereals, the most abundant protein fraction is the prolamin. These reserve proteins are synthesized in the protoplastids during grain development. The environmental conditions and soil fertility affects the amount of prolamins. In general, a high nitrogen fertilization level increases the amounts of prolamins that are stored in protein bodies distributed throughout the endosperm cells. They are water insoluble but have good solubility in alcohol. This protein fraction has different denominations according to the cereal grain. Rice, orzo, maize, wheat, gliadin, sorghum, kafirin, rye, secolin, and barley, hordine. From the nutritional viewpoint, prolamins are the poorest in terms of essential amino acid balance. These are rich in non-essential amino acids such as glycine, glutamic acid, and aspartic acid, and very poor in lysine and tryptophan. In some cereals, such as sorghum, the prolamins are highly cross-linked due to disulfide bonds. Therefore, reducing agents such as mercaptoethanol are required in order to improve the extraction rate. The glutalins are more difficult to extract due to their high molecular weight and presence of disulfide bonds. These proteins are extracted with detergents, alkalis, and/or mercaptoethanol. The glutalins are the main structural endosperm proteins. They are basically located in the protein matrix. The nutritional quality of glutalins is better than prolamins. In quality protein maize, the proportion of glutalins and albumins globulins are higher, and consequently the amount of prolamins is significantly reduced. Cereals contain high quantities of leucine, proline, aspartic acid, and glutamic acid. X component is lipids. Cereals have low amounts of fat-soluble compounds. However, lipids play an important role from the stability, shelf life, and processing viewpoints. The milling processes are aimed towards the removal of the pericarp and germ tissues. The selective removal of the germ or degermination is key because the oil rich in polyunsaturated fatty acids is highly susceptible to oxidation or rancidity. More than 80% of the total lipids are located in the germ. The maize germ is used commercially as raw material for the production of refined maize oil. The main reason is that the germ is comparatively larger compared to commercial cereals and efficiently obtained after dry and wet milling processes. These germs contain approximately 30–36% oil. Lipids are divided into saponifiable and unsaponifiable compounds. The saponifiable are subdivided into nonpolar triglycerides and polar monoglycerides, diglycerides, phospholipids, and glycolipids. Phytosterols, tocopherols, waxes, polycosynols, and carotenoids belong to the unsaponifiable fraction. The most abundant fraction is triglycerides because they comprise more than 95% of the total lipids. During malting or germination and respiration, the triglycerides are gradually hydrolyzed by lipases yielding free fatty acids. Faulty grain storage activates the grain, which synthesizes lipases that break down triglycerides, releasing free fatty acids. Free fatty acids are more susceptible to oxidation or prone to rancidity and therefore yield off odors and flavors. Most of the fatty acids that constitute the triglycerides are unsaturated, approximately 70% are oleic 18,1 and linoleic 18,2. Palmitic acid 16,0 is the main saturated fatty acid and generally constitutes 15%-25% of the total amount. The pololipids, phospholipids, and glycolipids are present in small quantities. However, they have important metabolic functions. X component is carotenoids and pigments. Carotenoids are polysoprenoids containing 40 carbons that are subdivided in two groups, carotenoids and xanthophylls. Carotenoids are the biosynthetic precursors of the oxygenated derivatives, or xanthophylls. These molecules are conjugated polyene chains that act as antioxidants. Carotenoids are very minor constituents in cereals grains. They are mainly present in yellow endosperm cereals such as yellow maize, yellow sorghum, and durum wheat. The maize gluten meal is especially rich in both carotenoids and xanthophylls. This co-product is widely used by the poultry industry to achieve the degree of pigmentation in egg yolks or broiler skins. The B-carotenes are precursors of the active form of vitamin A. Upon ingestion, the B-carotenoids transformed into retinol in the human body. The xanthophylls, zeaxanthin, and lutein are considered important nutraceuticals due to their implication in human health, vision, and eye integrity. The tocol derivatives, tocopherols and tocotrienols, are responsible for the vitamin E activity of plant tissues. Cereals can contain up to 8 different tocol derivatives, being the B-tocopherols and S-tocotrienols, which are most abundant in wheat, rye, and triticale. Conversely, the white tocol derivatives are present in maize, rice, and millets. Barley contains both SS and white tocol derivatives. Tocol derivatives are mainly located in the germ and therefore migrate with the crude oil after mechanical or solvent extraction. Next up is minerals and vitamins. Cereals are considered an adequate source of certain minerals and vitamins. The germ and aleurone layer contain the highest concentrations of these nutrients. The various types of milling systems yield refined products that contain lower amounts of vitamins and minerals. This is the main reason why these products are generally enriched with iron, zinc, vitamins B and B. Niacin and folic acid. Cereals are considered a poor source of calcium. Phosphorus is the macromineral found in the highest concentrations in all cereal grains. Most of this essential mineral is bound to phytic acid and phytin, potassium, and magnesium salts of phytic acid. Phytic acid has several relevant physiological functions such as antioxidant protection during dormancy, storage of phosphorus and cations, and serving as a precursor of cell walls. In addition, phytic acid plays an important and critical role during germination. Approximately 80% of the total phosphorus is bound to phytates in wheat, rice, and maize. Most phytic acid is found in the aleurone cells, although in the special case of maize, 80% of the phytates are located in the germ. The phosphorus bound to phytates has a low bioavailability, 40%-80%, and binds other minerals such as Ca, A, E, U, and Fe, lowering their availability. The availability of phosphorus and other minerals improves after germination or malting and fermentation due to the production of phytases. Most magnesium is located in the aleurone layer and is commonly bound to phytates. Therefore, most of the magnesium is lost during milling processes. Cereals are also considered an important source of potassium, but they are practically devoid of sodium. Most of the iron, zinc, and copper are in the pericarp, germ, and aleurone, and therefore considerable amounts are lost during milling operations. Cereals are considered one of the best sources of B vitamins, thiamine, riboflavin, niacin, pyridoxine, and folates, but a poor source of fat-soluble vitamins and vitamin. Most B vitamins are associated with the aleurone. The niacin associated with cereals is found in free and bound forms. The alkaline treatment of maize for production of tortillas and other traditional food products increases niacin bioavailability because the alkaline heat treatment breaks the glycosidic bond that binds free niacin with the other components.